In this section, we are going to be covering some general information when it comes to the biosynthesis of nucleic acids, more specifically DNA replication. So, one of the things that we must establish, and we have already mentioned in previous sections of the lecture, is that when it comes to DNA replication, in prokaryotes, these events happened in the cytoplasm, but when it comes to eukaryotes, DNA replication happens in the nucleus of the cell. Now, the process of DNA replication explains how from one double-stranded DNA molecule, we are able to generate two double-stranded DNA molecules. And we're going to see right now this general information of how different experiments prove how this process happens. So, when it comes to the flow of genetic information, we have already discussed it. When it comes to how the different nucleic acids form, resulting in the overall creation of proteins, it goes as follows. The molecule of DNA undergoes the process of transcription to make RNA. RNA, in turn, is going to be undergoing the process of translation in order to make protein. But when it comes to DNA replication, understand that this process is how we make more DNA. And in the case of bacteria, they copy their DNA continuously because they don't go through a well-defined cell cycle. In other organisms, like eukaryotes, like human cells, DNA replication is controlled within the context of the cell cycle, meaning that DNA replication is going to take place specifically during the synthesis phase of the cell cycle, and that synthesis phase is also known as the S phase. The replication of DNA faces three important challenges during the whole event. Number one, during DNA replication, we are going to be separating two strands and they must be protected from nucleases. Nucleases are enzymes that cut the nucleotide chain, meaning the backbone, in nucleic acids. Another challenge that is faced during this process is the synthesis of those new strands themselves. Remember, DNA is an anti-parallel structure, and having this directionality in this double-stranded molecule presents a challenge because we are only going to be synthesizing this DNA in the direction from 5' to 3'. And we're going to see how the system overcomes this challenge. Lastly, when DNA replication is happening, we need to be guarding if errors are going to be happening in the process. At the end of this chapter, we're going to see how the system not only proofreads, but fixes any mix matches that are going to be happening during this process. First, let's discuss how did science figure that replication itself is called a semiconservative process. So, the work for representing or showing that DNA replication is semiconservative were the experiments that were performed by Melsenson and Stahl in the 1950s. So the basic experiment go as follows. They hypothesize that DNA is going to replicate semiconservatively. So semiconservative suggests that in DNA replication, is produced two molecules of DNA, and they are formed or composed of a parental strand plus a new strand. And let's see how they came to this conclusion. So in the experiment, what they did is that they had a culture of bacterial cells that was grown in nitrogen 15 medium. What that means is that because nitrogen, it is utilized in the biosynthesis of nitrogenous bases, this nitrogen 15 will be now incorporated into the nitrogenous bases of the DNA of this bacterium. Now, they transfer some of that N15 bacterium into a flask that had a different isotope for nitrogen, which in this case was nitrogen 14. And they allowed the bacteria to grow. Then they were taking samples at different time points because replication for bacteria actually happens much faster than eukaryotic cells. And they took samples specifically at 0 minutes, 20 minutes, and then 40 minutes. At 0 minutes, what they were looking at is how does having just heavy labeled DNA will look like. And as you can see, the sample at 0 minutes demonstrated that the parent DNA was all heavy. When they explored what happened to the flask that now had 15 label isotope and then nitrogen 14 label isotope, meaning now we have bacterial cells that have the heavy label and the light label. When they looked at that first generation, they only observed that it was all intermediate. All intermediate means that the DNA had N14 label and N15 label. Then they took a second sample, as you can see, at 40 minutes. And when they saw that, they saw half intermediate and half light label. So the results illustrated that after 2 generations, half the DNA was intermediate and half was light only. And there was no double-stranded heavy at the end. So they concluded by having these observations that the pattern observed in the double-stranded DNA that they isolated has to come from the semi-conservative trend, meaning that there is a parental strand and a new strand. So these that I highlighted in yellow are what are resulting from DNA replication. Now, another thing that we need to consider during the process of replication is which direction does replication go. In prokaryotes, understand that there's going to be pre-replication complexes that are going to bind the ORI sequence. This ORI sequence is the origin of replication sequence. Now, what we must explain is that during the process of DNA replication in prokaryotes, what we see is that there are 2 replication forks formed. And these replication forks are going to be these ends where we have this double-stranded DNA being separated or unwound. And these forks are going to be moving away from one another. When it comes to eukaryotic cells, understand that there's going to be many ORI sequences or sites, and when the initiation of replication happens, understand that there's going to be replication forks on each of those bubbles that are going to be moving away from each other, and that's how replication is originated. When it comes to the enzyme that it is responsible for catalyzing the structure or for mediating the synthesis of these new DNA molecules, understand that DNA polymerase is what catalyzes the continual addition of each nucleophile that is resulting in the elongated polynucleotide that, for this case, we're talking about making more DNA. Now, understand that when it comes to the polymerases, we're going to have different polymerases for prokaryotic DNA replication and for eukaryotic DNA polymerization or DNA replication. Now, this list is limited, and towards the end of this presentation, you will see that the list of what we know to date is much larger. But in general, the most important DNA polymerases that are involved in this system for prokaryotic polymerases include DNA polymerase 1, and its job is going to be to erase the primer and fill the gaps on the lagging strand. We're going to have our DNA polymerase 2, which is going to be involved in DNA repair, and DNA polymerase 3, which is going to be involved in DNA synthesis itself. When it comes to eukaryotic polymerases, as you can see, instead of having them as Roman numeral 1, 2, and 3, we're going to have alpha, beta, and delta subunits of those polymerases. As you can see here, we also have what is their function, but understand that we are not going to be looking at DNA replication in detail for eukaryotes. We are going to focus our efforts on looking at DNA replication in prokaryotes. In order for DNA polymerization to occur, understand that there's certain requirements. This figure is representing a DNA polymerase reaction outside the cell. Meaning, this is when we are trying to make more DNA in the laboratory, these are the components. But understand that these components that we need for polymerization reactions outside the cell are very similar to the requirements that we have inside the cell. So, whenever polymerization of DNA has to happen or DNA replication has to occur, we need a DNA template. In the case of nature, that DNA template is going to be the parental DNA. We need a primer, and understand that in natural replication, our primer is going to be RNase based. We need DNA polymerase, and as I just illustrated, in the case of prokaryotes, we're going to have 3 types of DNA polymerases acting on that template in order to accomplish the replication of DNA. We're going to need deoxynucleotides because remember that we are dealing with making more DNA, so the sugar motif must be deoxy. And lastly, even in nature, we are going to have divalent cations present. For example, magnesium. It is known that in the case of magnesium, it is having a dual role in the system. The first role is going to be stabilizing the negative charge in the backbone of DNA. Remember that DNA has phosphate groups, that 3' to 5' phosphodiester bond. And then the second thing that has been demonstrated is that magnesium is important at the active site of DNA polymerase to help the catalysis that has to occur with this enzyme. Another feature of DNA replication is that it's going to happen semi-discontinuous. And sometimes we may wonder, why is it that this process does not happen in a continuous manner? So, one of the things that we must remember is that this has to do with the nature of synthesis of DNA by the DNA polymerase enzyme. What do I mean by that? It doesn't matter which strand are we trying to have this DNA being replicated. Remember, DNA synthesis always occurs in the 5' to 3' direction, because remember that the 3' hydroxyl group is our nucleophile and our 5' phosphate, the first one, is going to be our electrophile in the system. So understand that the reason why DNA is happening semi-discontinuously is because during the process, as our replication fork, which I just highlighted in green, is separating those strands, we need templates that are in the 3' to 5' direction. And as you can see, the template on the top has the correct directionality. It is already unzipped in the 3' to 5' direction. So that leads to the creation of a strand that can be made continuously. But the reason why this is done semi-discontinuously is because remember that the complementary strand of that original parent double-stranded DNA is going to be in the 5' to 3' direction. So when it is synthesized, we need to utilize the template in the opposite way. So when it's read, as you can see, because the synthesis happens 5' to 3', it must be read away from the replication fork. And that's why that second strand is made in pieces. As I explained before, synthesis of DNA always occurs in the 5' to 3' direction because of the following reaction. Every time we are trying to polymerize, these DNA molecules, making more DNA, understand that what we have is that the hydroxyl group at the 3' position, in this case, in our deoxyribose sugar motif, is going to bind to the incoming nucleotide. That 3' hydroxyl group is going to be the nucleophile. The phosphate group is going to act as our electrophile, meaning that it's going to now accept those electrons that are coming from the lone pairs on the oxygen atom in the hydroxyl group. And overall, this two phosphate groups are going to be leaving the system as inorganic phosphate. So they are the leaving group. And the result of this process is the formation of a 3' to 5' phosphodiester bond between, as you can see, the sugar motifs in that 3' to 5' phosphodiester bond, along with the sugars, are going to be what gives rise to the backbone of the DNA molecule.